In mammalian cells, the endoplasmic reticulum, or ER, plays an important role in protein biogenesis and calcium signaling. The heterotrimeric Sec61 complex in the ER membrane provides an aqueous path for newly synthesized polypeptides into the lumen of the ER. Recent data suggest that Sec61 may also form transient calcium leak channels. To directly address the role of the Sec61 complex as a potential calcium leak channel and to characterize its putative regulatory mechanisms, cells are first treated with siRNAs directed against the Sec61A1 gene. In complementation analysis, the cells are co-transfected with an iris GFP vector that allows for the siRNA-resistant expression of the wild-type Sec61A1 gene. Then the cells are loaded with the ratiometric calcium indicator, Ca2, and changes in the cytosolic calcium concentration are monitored via fluorescence microscopy. As the calcium levels are measured, solution changes are performed to assess the impact of various agents, such as calmodulin antagonists and thapsigargan, on calcium leakage. Quantitative analysis of the resulting images reveals changes in calcium efflux induced by the silencing of Sec61A1. In this video, a model for the role of Sec61A1 in calcium efflux will be presented following demonstration of the procedure and presentation of the results.
The main advantage of this technique over existing methods like planar lipid bilayer experiments or single-channel measurements is that candidate membrane proteins can be analyzed in their native environment and without tedious purification and typically inefficient functional reconstitution.
This method can help to answer key questions in the calcium signaling field, such as, for instance, what is the contribution of a specific ER membrane proteins to the calcium efflux from the ER in different cell types and how are they controlled?
Demonstrating the procedure will be Sven Long and Nico Schäuble, two talented graduate students from our laboratories.
Begin this procedure by preparing the solutions and reagents to be used. First, prepare calcium-free buffer according to the instructions in the accompanying written protocol. Next, dilute a stock solution of the calcium indicator dye Fura2AM into 1 ml of DMEM containing 10% FBS and 1% penicillin streptomycin to a final concentration of 4 micromolar. Dilute a 1 millimolar thapsigargan stock solution in calcium-free buffer to a final concentration of 3 micromolar. Also have on hand 200 micromolar ophiobolin A and 20 micromolar trifluoroperazine. To reduce the risk of studying off-target effects of a particular siRNA, 2 different siRNAs are used to silence each gene. To assess possible side effects of the transfection procedure, negative control siRNAs, which are composed of a random sequence of nucleotides, are transfected in parallel with the experimental siRNAs. It is important that the effective siRNA-mediated silencing can be rescued by expression of wild-type cDNA. Therefore, the siRNAs used for the experiment are specifically selected so that the rescue plasmid is not affected by the siRNAs. In this experiment, siRNAs are directed against both the coding and non-coding region or untranslated region, abbreviated UTR, of the gene of interest. When siRNAs are directed against the UTR, rescue plasmids without the endogenous UTR can be used. To perform transfections in HeLa cells, prepare the cells for the transfection by placing a 25 mm polyL lysine-coated cover slip in one 6 cm cell culture dish for each transfection. After 1 hour, add 5.2 × 10 to the 5th HeLa cells in supplemented DMEM to each dish. To transfect the cells, label 1 microcentrifuge tube for each transfection reaction. To each tube, add 4 microliters of 20 micromolar siRNA dissolved in 80 microliters of Optimem. Here, Sec61A1, Sec61A1 UTR, and negative control siRNAs are prepared. To each tube, add 20 microliters of hyperfect transfection reagent. Gently vortex, then incubate the reactions at room temperature for 10 minutes. To transfect the cells, add the siRNA mix, 104 microliters in total, dropwise to the seeded HeLa cells and gently swirl the dish to ensure uniform distribution of the transfection complexes. Incubate at 37 degrees Celsius in a humidified environment with 5% carbon dioxide. After 24 hours, replace the medium with 3.9 milliliters of fresh medium. Then, to increase transfection efficiency, perform the transfection a second time using the same siRNA mix. To evaluate silencing, perform Western blot analysis. Silencing should be above 80% as shown here. For Ca2 the maximum silencing effect is typically seen 96 hours after the first transfection. "48 hours after the first transfection, prepare to rescue the phenotype resulting from Ca2 silencing. Begin by labeling a new microcentrifuge tube for each reaction. The cells will now be transfected with either empty vector or wild-type Ca2 expression plasmid. The wild-type expression plasmid used here was generated by inserting Ca2 cDNA into the multiple cloning site of a pcDNA3 internal ribosomal entry GFP vector containing the cytomegalovirus promoter, the iris, plus the green fluorescent protein coding sequence. Prepare the transfection reagent by adding 4 micrograms of each plasmid dissolved in 80 microliters of Optimem to each tube. Then, to this, add 16 microliters of Fugene HD transfection reagent. Gently vortex this solution and incubate it at room temperature for 10 minutes. Then, after exchanging the medium, add the plasmid mix dropwise to the seeded 5.2 × 10 to the 5th HeLa cells and gently swirl the dish to ensure uniform distribution of the transfection complexes. Place the cells at 37 degrees Celsius and 5% carbon dioxide and incubate for a further 48 hours. To determine transfection efficiency, evaluate GFP expression by fluorescence microscopy after 48 hours. At least 80% of the cells should be transfected. Prior to imaging, use forceps to transfer the cover slips with the transfected HeLa cells to a new 3.5 cm cell culture dish and add 1 ml of DMEM with 4 micromolar Fura2 Incubate for 45 minutes at 25 degrees Celsius in the dark. To perform imaging, transfer the cover slip with the Fura2 loaded cells to a metal recording chamber and fix it into place by screwing the two parts of the chamber together. Using a pipette, add and remove 300 microliters of calcium-free buffer to the cells to wash them. Repeat. Then, mount the recording chamber on an iMic microscope with polychrome 5 chromator. Using a filter set for Fura2, find a field containing at least 20 cells. In order to estimate the calcium-independent fluorescence of Fura2, the excitation wavelength used in this step is 360 nanometers. Once a suitable field is selected, set up the imaging software to excite at 340 nanometers and 380 nanometers and to measure the emitted fluorescence at 510 nanometers every 3 seconds. Set the exposure time to produce fluorescence intensities around 1000 arbitrary units. For the setup shown here, a 5 millisecond exposure time is set. Additionally, mark the regions of interest, or ROIs, for a cell-free background region and up to 20 cell contours. Initiate live cell imaging. While the experiment is running, the system will present the ratiometric images for Fura2 signals as well as the real-time F340-F380 ratios for each marked ROI as a line chart, where F340 and F380 correspond to the fluorescence intensity within the ROIs at 340 and 380 nanometers respectively. After 1 minute, add test solutions to the imaging chamber to test different experimental parameters. In these experiments, the calmodulin antagonist trifluoroperazine and ophiobolin A are tested. After ratiometric measurements are carried out for an additional 10 minutes for calmodulin antagonists, add dapsogardin and continue the measurements. Record 400 paired images per experiment. Once images have been collected, review the images and mark the ROIs for at least 20 cells for each field of view. Subsequently, use the kinetic analysis option in the software to create a sequence of background subtracted ratio images and a plot showing the background subtracted calcium ratio kinetics for each ROI. Finally, export the data to Excel and Origin. Using an established calibration method, the cytosolic calcium concentration can be calculated from ratio measurements. To examine whether Sec61A1 gene affects calcium leakage from the ER, the Sec61A1 gene was silenced by 2 different siRNAs in HeLa cells for 96 hours. Silencing was evaluated by Western blot analysis of similar cell numbers in triplicate. As shown here, there is a strong Sec61 alpha signal in lanes 1 through 3 and a significantly weaker signal in lanes 4 through 6 and 7 through 9 that correspond to the Sec61A1 silenced cells. To quantify the results, the Sec61 alpha signals were normalized to beta-actin signals. Greater than 80% silencing was achieved. To test the role of calmodulin in ER calcium efflux, Fura-2 imaging of Sec61A1 siRNA-treated HeLa cells was performed in the presence or absence of the calmodulin antagonist ophiobolin A. This movie shows control cells treated with ophiobolin A. 10 minutes after ophiobolin A treatment, calcium release was initiated by applying thapsigargin, a SERCA inhibitor that unmasks calcium release from the ER in the absence of external calcium. As can be seen here, the addition of thapsigargin led to a transient but significant increase in fluorescence, indicating calcium efflux from the ER. In contrast, calcium efflux following the addition of thapsigargin in ophiobolin A-treated Sec61A1 silenced cells is much less pronounced, as can be seen here. This difference can be seen clearly in this diagram, which shows estimated cytosolic calcium over time in a series of movies. The quantitative results shown here confirm increased calcium efflux in control cells in response to thapsigargin in the presence of ophiobolin A. As can be seen in this bar diagram, the same phenomenon was seen for a second CAM antagonist, trifluoroperazine. However, the effects of the CAM antagonists were not seen when Sec61 complex was depleted from the cells by either one of the 2 Sec61A1 specific siRNAs. Partial rescue of this phenotype can be seen when Sec61A1 silenced cells were transfected with Sec61A1 expression plasmid. As can be seen here, calcium efflux in response to calmodulin agonists is partially restored with transfection of the siRNA-insensitive wild-type plasmid. These results suggest that calmodulin is involved in limiting ER calcium efflux at the level of the Sec61 complex in cells. If so, transfection of Sec61A1 gene with a double mutation in the calmodulin binding site should not rescue the siRNA-induced phenotype. Indeed, when cells were transfected using a Sec61A1 expression plasmid with a double mutation in the IQ motif, there was no rescue of the phenotype. Taken together, these data indicate that the release of nascent chains from the Sec61 complex indeed leads to calcium release from the ER and the formation of a calcium nanodomain around the cytosolic mouth of the Sec61 complex. This calcium is bound by calmodulin, and calcium-calmodulin closes the Sec61 complex.
After watching this video, you should have a good understanding of how to combine siRNA-mediated gene silencing in human cells with live cell calcium imaging.
Following this procedure, other methods like the expression of cDNAs with disease-linked mutations can be performed in order to answer additional questions, like the potential impact of disease-linked mutations in relevant components.
And don't forget that working with drugs such as thapsigargin can be extremely hazardous, and precautions such as the use of gloves should always be taken while performing this procedure.